Okay, well, my name is Maurice Maloney, although you might mistake me with this voice as being Don Corleone from The Godfather. I'm just doing a little bit of rehearsing for that role. But we've got a very exciting set of presentations for this morning. While I'm up here, I just want to say a word of thanks both to Summit organisers and folks at ULIC and other places as well that have been involved in this. This has been a really good symposium so far, and today is going to be excellent as well. So really a big thank you to all of who've been involved in the organisation. So I'm going to be co-chairing with Gemma Molero, who's sitting over here. So we're going to tag team a little bit to introduce the speakers. And our first speaker this morning is Michelle Watt. And I think Michelle is not a stranger to most of the people in the room. She's co-director of plant sciences, and if anybody continues talking, I will act like the Godfather, so smack hand, okay? So let's calm down a little bit. So Michelle is co-director of the plant sciences Forschungszentrum in Jülich in Germany. She's a professor of crop root physiology at the University of Bonn, and she also leads the root dynamics group there, which focuses on root and rhizosphere discoveries using advanced phenotyping technologies. I've known Michelle for quite a long time. She was one of the stars in CSIRO in the agriculture, what was called plant industry in those days, and developed a very consistent research program looking at all of those things that plant breeders normally don't see. That is all the secrets hidden below ground, but which make a massive difference to crop yields. So Michelle's talk today is phenotyping for root-based gains in crop productivity.
Okay, I just actually have to find my talk here. Okay, thank you, Maurice. I'll start my slide with an overview of an example of root traits that actually have reached some part of a breeding process. This is basically to say that although roots are below ground and difficult to study, there's actually been a reasonable amount of effort and one might describe in the context of traits generally, success towards delivery and impact in breeding. Starting at the bottom, one of the first efforts was actually conducted in Canada by Hurd and colleagues. He actually used rhizoboxes to identify wheats with a deep root proliferation for better water use in prairie systems. And then he just used the information from those rhizoboxes to allow those genotypes to become parents in breeding. Subsequently, Matthew Reynolds and colleagues have used canopy temperature as an indirect surrogate of likely deep-rootedness and deep water capture, actually within selection in breeding. And more recently, Anton Wasson and CSIRO colleagues and I used field coring to identify deep-rooted wheats, and these wheats went into as breeding parents in India. There's also been success in identifying single gene markers for cereal cyst nematode resistance, and of course root disease tolerance has a massive impact on water and nutrient capture. Aluminium tolerance, a major mechanism of malate release and keeling aluminium around root tips, was discovered and translated into markers by Delhaize, Peter Ryan, and other colleagues at CSIRO. Similarly, a sodium exclusion transporter also developed into a marker. And then there's actually been a range of activity around root angle. First, Jonathan Lynch's group looked for a shallow root angle in bean to acquire surface P, particularly in South American and Chinese cropping systems. More recently, there's been the discovery of the DRO1 gene, a gene associated with deeper rooting in rice by Uga and colleagues, and Graham Hammer and his team in Queensland have actually been working for some time in a root angle trait in wheat, and this is also making its way to a marker for breeding. Way back in the 1970s and 80s, John Passura and Richard Richards did an anatomical selection for narrow xylem angle. And for phosphorus efficiency, Matthias Wissewa and Sigrid Hoyer identified genes in the PUP region, so single genes associated with nodal root initiation under low P. So the features of these activities are that they've all involved phenotyping for selection, either starting in controlled conditions or starting in the field and possibly combining the two of them. Shoot phenotypes have been a part of the selection. So far they're all single traits, either polygenic or single genes. They either are a functional or developmental trait, and they've made different extent of sort of penetration into actually germplasm improvement. Really, these have been in public breeding operations, mostly pre-breeding activities, and have had quite long delivery times, but not particularly long compared to shoot examples that I'm sure Greg will talk about later. So the question is, where can current and future phenotyping technologies progress root-based crop improvement? And there is an interest and a demand in continuing, obviously, to look for root trait-based improvements. And I've listed here a range of these demands, and in my talk I will cover those, giving these examples alongside where technical opportunities have opened up. And there are also phenotyping solutions to extend root-based phenotyping with modern phenotyping technologies. And I'm going to start first with dynamic or functional root traits, and we've heard a little bit about those potential traits already in this meeting. Basically, dynamic or functional phenotypes in roots could be traits where the roots initiate a growth process or a functional process in response to a change in the soil environment. So, an example, of course, is water, a rainfall event, and water becoming available. And the sort of technologies that can track these processes, one of them is the use of the magnetic resonance imaging technology. This is where root systems can be imaged directly in soil, and Mark from the Fortune Centrum also introduced this technology yesterday. This is a maize root system in soil. It's been imaged over time. The image analysis is directly extracted from the images in the cores, and with those time-lapse images, he's actually illustrated the rates of growth of the different roots on the root system with these colored balls. And there's some very interesting developmental and physiological aspects that can be seen in this image that I won't get into. One of the main things is not only do roots elongate, but they actually stop elongating, and I won't go into that too much more, but this is a very interesting aspect of root architecture that we don't really understand. So here's the MRI system here that Mark introduced, and the key aspect to it is that we currently have a small robotic delivery system to put plants continuously overnight in and out of the MRI, and this is actually in the process of being extended and linked to a controlled environment chamber. And the example I'm going to give is looking at maize in pots that are 8 centimeters by 40 centimeters deep. Here's the data here from an experiment conducted by Robert Kohler, basically looking at 2 scenarios, one where root systems of wheat continually received water, and the other treatment, the watering was stopped, so a drought period was imposed, and then the plants were rewatered, and imaging was carried out in time points in the 2 treatments. We're seeing here what we've already seen a little bit about in this meeting. Stefan Girth mentioned this in relation to potato, for example. There seems to be a situation that when the roots are rewatered, there's this increase in growth rate compared to the control. The total root length has actually been extracted directly from the MRI image root system, so there's no washing involved, and we can extract much more spatial information from these images to go in and look and see what parts of the root system are actually responsive to this rewatering. And I won't talk about this today, but Robert also is using N15 labeling to get additional functionality associated with this drought responsive that's imaged with the MRI. And in a second example from the MRI, this is a study where the Jülich scientists modified the design of the tube that goes into the MRI so that many smaller plants, in this case, they were actually working for a horticulture company that was interested in robust, rapid, and drought-tolerant adventitious rooting from the stem of petunia cuttings. So they had much smaller plants, and they were able to design this tube so they could stack the plants and using the robotics, repeatedly have them go in and out of the system. So actually the throughput of the MRI is starting to get up there in terms of numbers. And here what you can see in the images is not only are the roots imaged, but so are the shoots. And here, what you couldn't focus on in this image, here's the cutting, and in this area right here is where the roots develop from the stems, and these 3 genotypes have a difference in their ability to root. What they also did, Dagmar specialists with the MRI, is going on to use not only the water signal in the roots, but the water signal in the shoots and the water signal in the soil to look at short time course events in terms of what's happening upon rewatering within hours. So this is getting away from developmental responses into hydraulic responses and genetic differences in the sort of things that Xavier Dre was mentioning yesterday. And this is, on these axes here, this is the amount of water on the y-axis, and this is the soil water loss, so he can track the soil water loss from these 2 genotypes. This is the plant water loss. So here's the change in the water movement into the shoot. Here's the change in the water movement out of the. Sorry, I'm going to start this again. This is one genotype, and this is second genotype. This is the water movement moving into the root, and this is the water movement into the shoot. So in this genotype, the root is slightly ahead of the shoot. In this genotype, the slope is less, and the change between the tissue movements is seen a little bit later. And similarly, this genotype here draws water more quickly from the soil than this genotype here. And these are phenotypic selection methods that can start to look at hydraulic, quick responsive differences between plants. So now I'm going to move to the next area, an interest with field phenotyping, and that's linking to field traits. I'm going to jump immediately to basically describing the two direct phenotyping methods we have for roots generally among research groups around the world at the moment. And really, what I'm talking about here is direct root measurements. I mentioned an indirect method, for example, canopy temperature, that Matthew used. So there are indirect methods, but these are methods where people are directly looking at the roots. There are two methods. One involves a soil core pushed into the ground, and the other involves the shoveling of the root crown. And this is the method developed by Jonathan Lynch and known commonly as so far as shovelomics. The main thing is that they get two very different types of data. The core data is a root count or root length density down the profile from a core face. There's no washing involved. You can potentially get a very deep measure to 2 meters, and you're getting this transect through the root profile. The shovel data is a crown, is the root crown. It's washed. It's a shallow parameter. It has the advantage of being associated with the stem in a plant, and then it's imaged. And recently there have been automation advances with both of these techniques. With the crown root phenotyping, at ULISH we've developed a box that standardizes the light environment for imaging the crowns after they've been washed. It's got a little motor and it rotates the crown so that there can be a complete collection of 25 images around the whole crown, avoiding this problem of deciding which sides to image. And currently we're mainly using the DIRT image analysis tool developed by Alexander Bush, but we're developing a new in-house tool to capture more parameters from these rotating crowns. The good thing about this is that it's portable, and also it can be adapted to different species, and we have currently adapting it to cassava for collaboration for doing the field phenotyping directly in Thailand. In terms of the coring, is probably done still most intensively at CSIRO. This is an example of the corer that's at CSIRO. It's attached to a tractor. It's got a hydraulic mechanism to push the core into the ground. I understand that there's a corer, similar one at CIMMET, I think, and really there's it's very possible to build such a system. It obviously makes going deep to beyond a meter to 2 meters to look at roots much more feasible if you have a corer. Anton Wasson and colleagues at CSIRO also developed an imaging box for the cores. This is a portable box that also goes to the field, so no lab processing. The cores are mounted in this box. This box also has special fluorescence type illumination to intensify the difference between the roots and the soil. There's an automated image analysis program developed for these core faces, and this is all published in this JX Bot paper. So there are advances with direct root phenotyping for crowns or for deep cores. Now I'll move to the next interest that we've been hearing about in this meeting. This is probably particularly related to the nice modeling talk that we heard from Fred after lunch yesterday, and this is really trying to integrate phenotypic data into models to help understand root by environment interactions. And what we've done in ULISH towards this objective is we've taken the SimRoot model, which is a structure-function root architecture model developed again by Jonathan Lynch's lab over quite a number of years. This model was actually written and developed by Johannes Posma, his PhD student at the time, and Johannes has been at ULISH for about 5 years, and Johannes and Christian Cooper, who's actually here at this meeting, have been rewriting this code. It's now available online. We open sourced it, and it has extended parameters that I'll talk about now. So one of the functionalities that Johannes and Christian have added to the model, now also in collaboration with Malcolm Bennett's group in Nottingham, is to be able to use the SimRoot model with phenotypic data from either the NMR, these are MRI-acquired images, or from images acquired from CT. These images here were collected by Darren Wells, who's also here at this meeting. And SimRoot can take this information from these two non-invasive methods. It can actually aid in the extraction of the data from the images, and once that data is in the model, we can start to use it to understand and test functionality. And in this image here, this is the root system in SimRoot here from the CT data. This is the root system from SimRoot from the NMR data, so you can see they're very well represented. And in this image here, a nitrate function zone of exploiting has been overlaid using the SimRoot model. So the Open SimRoot model, this is a functional structural plant model. It has an emphasis on root architecture, but it has a simple radiation use efficiency shoot type crop model, and it also has it's also been parameterized for carbon, basically the exchange of carbon for nitrogen and phosphorus resources. So it accounts for carbon fixation and photosynthesis, and then whatever is not used for shoot growth goes down into the roots. And it has an explicit root architecture model, so it allocates this carbon into different root classes and types. And then it is parameterized for responses to the nutrient concentrations in the soil and what's available to the plant. Christian Cooper and Johannes have now coupled it to a 3D soil model. And yes, they have been working a lot on parameterizing it for hydraulic. Yeah, so now I'm going to move on to what I think is an important aspect of root phenotyping, which is linking shoot traits to root traits. And this can really be done now in phenotyping platforms that are available in different locations, where shoot measures and soil or root measures can be phenotyped at the same time. And this is an example of a study done in ULISH led by Fabio and his group. This is a platform where plants are moved into an imaging chamber. Various images of shoots are taken. There's also a gravimetric capability that the plants are delivered to. And also we have microwave new technology. This is a scanner that scans down from the shoot into the root, non-invasively picking up water content in the shoot and the root. And he studied a small set of durum landraces and cultivars under 2 soil moisture conditions, measuring all of these parameters repeatedly over time. And what he found was a little bit like what Francois Tardieu and his team have been finding, is that there are a suite of traits associated with the shoots and the roots that are associated with different types of water use that combine shoot and root water phenotypes. He classified in this paper the germplasm into either spending types, a conductance type, an area type, based on leaf area, and a water-saving type. So these platforms can pick up a range of phenotypes that potentially can go into genotypic and phenotypic prediction for genotypes. The reason why measuring shoot and root phenotyping types simultaneously is so important is the fact that they're not distinct from each other. Root processes, phenotypes, they're lumped into generally either root growth and architecture processes, and this is where they actually colonize soil volumes to capture soil resources, or they actually carry out transport and signaling processes that influence and modulate shoot growth, and in particular can be very important for the harvest product. And we saw that a little bit about with the silk growth yesterday. So shoots and roots are tightly coupled. In a second experiment at Jülich, we demonstrated this for nitrogen use. This is using the rhizoscreen system. Many of you people are probably familiar with this. This is an automated, roboticized system of very large rhizoboxes. They're actually about this big. Again, they're moved into an imaging station. Roots and shoots are imaged over time. And what Kirsten and her team found in this study that the shoot and root traits all varied differentially under lower nitrogen supply. So this is a normalized dashed line for optimum nitrogen supply, and this is the response of the different shoot and root traits in the germplasm to lower nitrogen supply. And the main thing is that possibly there could be gain faster by shoot and root phenotyping simultaneously. And then finally, I'm going to go back out to the field, but still on the theme of the importance of linking shoot and root traits. I guess a small mention here about the technologies in the field. We're really only at the limits of destructive phenotyping, and this is a technology area where shoot sensors and hopefully root sensors can be discovered. This is the cutting limitation for technology advancements at the moment. But using destructive measures, the main two messages that I'd like to give from studies in the field where we measured shoots and roots. The main thing is that younger plant stages cannot predict later plant stages of roots. This is actually more a problem of developmental events that occur between young plant stages and mature crop root stages. Because even when you compare young plants in the field compared to older plants in the field, you get this nice relationship between early stages and still vegetative stages in the field, but then at flowering the relationship breaks down. So this is one thing to keep in mind, that young seedling stages, we cannot expect them to reflect the field at mature stages. And similarly, shoot phenotypes in the field, based on Alan Severini's PhD thesis, there are some nice sort of general relationships between shoot biomass and grain yield and rooting depth and root length density in the field. These are all generally positive, but they highly depend on soil environments. And in this example here, this is a range of material with different shoot dwarfing genes. You can see that shoot biomass and grain yield and rooting depth are not necessarily related. So with that, I'll finish and acknowledge my new colleagues at the Fortune Centrum in Munich.
Michelle's done a great job of staying on time, which means we've got time for a couple of questions. So, François. Oh, yeah. Yeah. That's right. This one's on.
Yeah, it's about the picture you showed on the fact that roots stop growing during water deficit. To your understanding, was it because of a mechanical impedance, or did the roots really lost the capacity to grow, or perhaps we just don't know?
This is actually something I don't know right now to answer that question, but Robert is able to, he's going in and looking at the MRI images now, excuse me, and because he can actually track the exact roots over time, we'll be able to see whether is it the same ones that stopped and then initiated, or was there actually new branch root growth coming from those, all of those things, because we have that spatial and temporal information. So this is quite new work, and he's continuing to analyze it. Yeah. Yeah.
Well, my question is very fast. Michelle, what do you think really about the shovelomics? Is really is really working?
Yes, so we have the crown root phenotyping running at ULISH. Tobias Wojciechowski has been using it for 2 or 3 years with different cereal crops, and he's finding genetic variation, and he's looking now at repeatability and heritability. Of course, Jonathan Lynch and Hannah Schneider, who's here, has been using it for maize in the U.S. and finding candidate regions.
My question is because at CIMIT they tried to implement Samuel Trachel was here, and apparently the results they got were not really very promising. That's my point.
Yeah. Look, it depends a lot on the soil type. There are many steps that need to be standardized because there's a washing step. And I think the automation of the image capture is helping for some of that variability. It really depends what you're interested in. Neither of them are, yeah.
Okay, Matthew Reynolds. Michelle, great talk. I just wonder, what is your, I'm going to give you a broad question here. What is your position on the extrapolation of root studies in pots to the field? And especially in light, I remember when I visited Jülich, seeing some great data that showed that roots are actually in pots extremely sensitive to their environment, and it has big implications for the way that the shoot responds. So, what's the utility? What's your position?
This is a good question, and actually we're discussing this quite a bit in the root phenotyping working group. A short answer can be shown in this panel here. This paper actually compares lab and field phenotyping, this panel. So basically young lab screens, even done in these paper rolls in wheat, at these same plant stages in the field, okay, directly harvested this way, not by shovel but by core and then washing, has a very good relationship between young labs in the field. In this example, in this field, it's at the mature plant stage that I really think we have to have a lot of caution about lab and field correlation. And like I was saying, this lab-field correlation has a lot to do with even plant stage, because here's the young plant stage, field to field, and here's the adult plant stage. This is actually one of the few examples of studies where people have actually compared lab and field. There has been very little effort to compare lab field. On that note, I don't think comparing lab and field is always the necessary activity in your pre-breeding. Field phenotyping has a couple of main roles. One is to maybe to validate. Yeah, this is such a long question, Matthew. The bottom line is it depends. and read the paper out of the root phenotyping group. It depends. And I think in some cases, controlled environment phenotyping is what is needed, and then a very final end, maybe field phenotyping of just two contrasting lines to like for example in your canopy temperature, it would be nice to validate at the end whether the roots were deep or not in a couple of germplasm examples. Yeah, great. So that's the thing. Field phenotyping must be in our armory of activities. Whether it's the right method for a high throughput selection for a genomic region, you have to assess this within your trait. Field phenotyping so far from quarrying and shovelomics is very, very variable.
So I think that question was worth two quick questions, and then we're going to have to move.
Hi, so Vincent Valdez. You talk about, linking shoot and root phenotype, but purely from a sort of architectural sort of standpoint. Would you be interested to look into the possibility of linking shoot and root phenotype from an hydraulic standpoint? And how, certain aspects of the root hydraulic could explain certain things that we see at the shoot level?
Yeah, we're very interested in that. And actually, in the Petunia example that I gave in the MRI, that water MRI information is giving us hydraulic processes. So we're already phenotyping for those with our developmental traits. And similarly, in the platform that I showed with the durum wheats, the work that Fabio did, also we are associating water functionality with the architecture traits. So we're definitely interested in that, and we're coupling those in the SimRoot and the Opensim root model.
Michelle, thank you for the talk. My question is really linking the last two questions. It's about field and shoot to root ratio. I think it's the shoot root ratio is very species specific. It depends what species you're working on. I give you an example, is perennial ryegrass that I showed yesterday. It can have very good shoot and very shallow root, or white clover as an example. But you have another clover, it's called Trifolium uniflorum, which has very small shoot and very deep root. So it's very much depending on species and also environment. You take the same species to different environments, and in dry environment you see longer root sometimes as a reaction to go deeper, and a small shoot for efficiency. And you go to a temperate environment and you see the other way around. What is your comment on that?
Well, I completely agree. So, and not only it's species specific, it's developmental stage specific, it's soil environment specific in terms of the transient and dynamic arrival and change of roots, for example, in the soil profile. And of course, I completely agree with Francois' comment yesterday that all these traits, for example, the ones that I reviewed at the beginning, only have value, trait value, in the conditions under which that trait is functioning for the plant. I personally am not a huge fan of root shoot, that word root shoot ratio anyway, but it's just further that what you've described there is the justification for phenotyping both, and that's what these new technologies are able to start to achieve.